I'd like you to imagine that one evening during rush hour, a collision occurs between two cars on a highway. Two cars quickly become three, and soon after, a multi-car pileup has occurred. Police, firefighters, and ambulances rush to the site of the accident, leaving nearby cities with far fewer first responders than they would normally have. Meanwhile, cars come to a standstill, and traffic stretches in all directions for several kilometers from the site of the accident. So what does this have to do with my research? Well, the interconnected highway system that you're imagining is actually very similar to the complex architecture of the brain. Networks of interconnected neurons transmit information between one another, and in rare and unfortunate circumstances, a genetic mutation can occur in a single cell. Much like the initial accident, as that cell grows and divides, it forms a population of cells with the same mutation. Now, that mutation gives those cells the characteristics of a tumor, and as that tumor grows, it draws on resources, blood, oxygen, and nutrients, moving them away from nearby healthy cells and impairing the ability of those cells to function normally. And as we saw with the first scenario, cancer becomes woven so tightly into the delicate nature of the brain that it's nearly impossible to remove. In fact, 9 out of 10 brain cancer patients will experience tumor recurrence within 7 months of having their cancer surgically removed, and fewer than 5% are alive 5 years after their diagnosis. The nature of my research is to understand how cancer cells spread and invade brain tissue on a molecular level. And our lab has recently discovered a gene that may be essential to just that. We have found that this gene acts like an on-off switch. When it's on, it activates signaling pathways that stimulate cell growth, division, and importantly for my research, cell movement. When it's off, cells can still grow, but they don't have the same capacity to migrate through the brain. Our research has shown that if we take brain cancer cells from patients at the Ottawa Hospital and we reduce their levels of this gene, we actually see these cells moving more slowly and migrating less. But cancer cell movement depends on a number of pathways that are acting together to stimulate the cell. So rather than pulling on the emergency brake, our approach so far has more so been to ease up on the gas pedal. Now, in my current research, I'm using a method of DNA editing called CRISPR. And with that method, I can remove this gene entirely from these brain cancer cells. And in doing so, I expect to see that the cells don't move as fast and don't spread as far, resulting in smaller and more confined tumor growth. And importantly, I'll be able to see what other aspects of the cell change when we remove this key regulator of movement. But brain cancer treatment hasn't changed much in the last few years, and I hope that by better understanding these pathways, we can jump the hurdle from cancer invasion to cancer cure. Thank you.